Hello, I am Pan-Jun. I'd like to thank Doctors Carl Tesser and Adam Craig for giving me this wonderful chance to talk here. If we look at the history of science and technology development in the world, then remarkable achievements like gunpowder, paper, and compass inventions were made in ancient China. But later, from some point, the true scientific revolutions of the modern sense happened in Europe. Famous science historian Joseph Needham, he asked the question, why did such scientific revolutions happen in the West, but not in China, despite its early success in technology inventions? And the typical answer to this question is that in the case of China, the centralized government system has been established so early. But in the case of Europe, it was relatively very much segregated. And as a result, the, the diversity of the ideas from here and there and its communications provided the foundation necessary for the scientific revolutions that we see. If I translate this idea to another form, imagine the fitness landscape of technology innovations in a given society like this. In this figure, each position on the plane corresponds to the condition of a given society. And the height from height at the position corresponds to the level of technology or science achieved by the society. In the case of technology development driven by centralized force, it may promote rapid climbing of technology development along the hill, but may rarely allow its movement downward from the hill. So probably the technology levels may stay near the local peak on this landscape and may not allow really its movement downward towards a higher and global peak on this landscape. But in the case of the technology development driven by diverse ideas rather than the centralized force, then there may be sometimes some suboptimal motion, like the movement downward from the hill, like in this figure. But that kind of rather suboptimal and random behaviors may eventually allow its movement towards a higher and global peak on this landscape. In other words, I suggest that the diversity-driven technology development may benefit the more fundamental breakthroughs in technology innovation at the long term. But these days, the centralized force for technology development, I think, may involve, the policies of the funding agencies, or the publication pressure from the institutions, and also as a main subject here, the commercial pressure from the industry for our research. In the case of an industry, they pursue the short-term returns for the short-term profits and also, the risk aversion, that kind of a commercial pressure maybe could, in some sense, for the technology progress, but may not be necessary, at the long term its development. For example, there is a report that in the case of pharmaceutical firms, such a commercial pressure in the face of the regulations, and they want to, avoid, they want to, the prioritize risk aversion, so actually that kind of a behavior suppress the novelty in their developed drugs. And also, I know the story very well. In the case of a biofuel research, in the mid 2010s, there was a sudden funding cut from some major company which supported biofuel research in some leading universities due to the due to some affair regarding the company and also the shale gas boom and the resulting oil price drop. So as a result, I know that the biofuel research in these universities suffered a lot from this funding cut. In this work, I like to particularly focus on the case of the effect of commercial pressure on generic research because I and my collaborators are actually familiar with the generic research than the other areas. Okay. First of all, we checked the funding sources of the research published in papers and also the assignees of the technology inventions in patents. And we found that in the case of most of assignees in patents, they were companies. But in the case of the funding sources of the papers, they are actually governments or some public foundations, etc. So we consider the technology inventions in patents as a proxy of industry-driven research, but in the case of the research in papers are not. Particularly to measure the research activities for a given gene, what we did is that we counted the number of the name of that particular gene in the titles and abstracts of the papers or in those of patents. And in the case of patents, we used the US, China, and European patents. And in the case of papers, we used the papers from various biological and medical journals. These graphs shows the examples of the trajectories of such numbers in papers and patents for some particular genes, like hemoglobin and ACE2. ACE2 is COVID-19 related genes, and here blue is the number in papers and red is the number in patents. And as you can see, they are skyrocketed at the time of the start of COVID-19, as we can reasonably expect. And over years, we identified which genes were very popular for the research in these papers and patents, like the papers here and patents here. And we counted the number of new genes studied per each year. So on the left side, you can see that there is an increase of the new genes studied in papers or patents until the year of around 2000. But after that, it started to decline. So we conclude that after the end of Human Genome Project, because it was year 2000, around 2000. So the study of new genes, it start to decline. But in the case of not the study of new genes, but instead the study of new combinations of genes, the situation is different. If you look at the graph on the right side, it keeps increasing, until some point, and after that, at least it does not decline. It stays still active. And I will revisit this result later in my talk. We found that in the case of industry-driven research for each gene, it drops earlier than the general, the scientific research driven by non-industrial sectors. For example, here, the gene COX-2, you can see that the patented, the research in patents, it drops early, around the year of 2004. But in the case of research in papers, it's not. And actually, not only COX-2, across all the genes, more than 60% of genes, as you can see the graph on the right side, the the uh, the industry-driven research is dropped earlier than the scientific exploration driven by the non-industrial sectors. And according to our case studies through COX-2 and PD5, PD5 actually famous for its related drug, Viagra. I believe that you know what is Viagra. And we know the reason from our analysis why their studies are dropped earlier in the case of industry-driven research. In the case of COX-2 and PD5, COX-2 2004, actually what happened is that the drug related to COX-2 was withdrawn from the market due to its serious side effect. But in the case of PD5, the reason is a little bit different. In the case of PD5, the drug for the PD5, it was approved not only for its original purpose, but also for the other diseases. So actually, these two cases respectively may be related to the industry-driven early drop of their research may be related to the risk management and also risk management purpose and the anticipated lower returns of their research investment due to the saturated market related to its drug, respectively. Interestingly, in the period of continuous scientific exploration, even after the drop of industry-driven research actually involve, the ongoing continuous innovative attempts in their research, like clinical trials. And if you remember, the active the study of new combinations of genes, as I already presented here, it suggests that we can use such new combinations of genes as another example of the innovative attempts regarding those genes involved in this combination. Actually, we found that the frequent genes, the most frequent genes in these combinations are the genes those represent the biotechnology innovations at that particular time, like the beta-galactosidase for DNA cloning, or the Cas9 for genome editing, or the GFP in the middle here, for the live cell imaging, and PDA1 for cancer immunotherapy, and so on. So we look at how frequently the genes are actually paired with these kind of genes, even after on the period of continuous scientific exploration, even after the drop of industry's research. And we found that the portion is quite substantial. So we asked, what if such a long-term scientific exploration is disrupted due to its over-reliance on industry's support? This was our question. And according to our estimation, at least a quarter of such innovative opportunities to be paired with such genes, it can be dropped. And in the case of some particular genes among them, even up to 70% of such opportunities would be gone. And consistently, we found that among many subfields in genetic research, the the fields of particularly high practical or commercial focus, like metabolic engineering, for example. Metabolic engineering is the field that researchers manipulate biochemical pathways of a given organism to increase the production of a given bioproduct. And those fields are actually, we found they show very low level of such, the, the the innovative attempts with the new genes for biotechnology innovation, and shows the overall lower level of the research vital innovative research vitality. This is our assessment. These days, the technology developments like AI or mRNA vaccines and quantum computing are dominated by industry, as you know. And but our work supports the stable, the course for the stable support for the long-term exploratory research in balance to preserve the long-term innovation vitality. This is our suggestion. Finally, I'd like to thank my team members and collaborators, and thank you for your attention.